This video is Transporter and Channel Fxn 1. So as usual for this course, we're going to spend most of our time zoomed in very close looking at cellular and molecular processes. But I want to remind you that these very small events which are taking place have a huge impact, potentially, on the whole organism. In particular, transporters and channel proteins are crucial for maintaining proper homeostasis, as well as allowing certain cellular processes to occur. This means that if these transporters are functioning incorrectly, it could have a huge impact on the organism, potentially leading to death, or otherwise impacting the health of the organism. In some cases, we've developed drugs which will maximize the function of some of these incorrectly working transporters, and so we'll hear some more about those later. In some cases, we know of specific diseases which are caused by malfunctioning transporters, and drugs have been developed to help with these processes, potentially helping these incorrectly functioning transporters to function more efficiently. And here's a couple of the highlights of the terminology and learning outcomes that we'll focus on and kind of build towards. So passive transport is going to be a key for this one. We're also going to talk about how each cell has a characteristic set of transporters. And one of the main highlights is the glucose transporter. This last part here about comparing and contrasting transporters and enzymes, I expect you to use your previous knowledge about enzymes to be able to answer this. So to define transporters, these are proteins which move water-soluble organic molecules and some inorganic ions across cell membranes. There are many different types of transporters, and these can vary with a particular cell type and or the organelle membrane within those cells or across a single cell. And each of these transporters is highly selective and specific. So looking at this figure, here's a couple examples. Basically, you can see that there's a cell that's drawn here, and that we're focusing in on some different membranes. So first off, we're going to start off by looking at the plasma membrane and the different transporters that are highlighted here. So basically, let's take a look at those. So for the plasma membrane, in this particular cell, it's highlighted that they have transporters which can move certain nucleotides, certain sugars, certain amino acids, and also that moves sodium and potassium across the membrane in a directional fashion, and that's the sodium-potassium pump. Another example is the lysosome, and so as a reminder about the function of that organelle, so the lysosome is the garbage disposal or recycling center of the cell, and so they will take larger molecules and break them down into smaller parts, and one of the main ways that this can be accomplished is through the proton pumps. So proton pumps here are going to bring protons in. That increases the acidity of this compartment, and that facilitates the breaking down of larger molecules, denaturing proteins, things like that. So that's a proton pump in the lysosome, which increases acidity and assists in the breakdown of large molecules. I also want to highlight the mitochondria, and in particular here we're looking at one mitochondrion and looking at the transporters which are found in the inner mitochondrial membrane. So two that are depicted here there's a pyruvate transporter, which you may recall from an introductory biology course. So pyruvate is a large-ish organic molecule, and so there's particular pyruvate transporters which will bring pyruvate into the mitochondrion and then allow enzymatic reactions to occur within the mitochondria. There's also a ATP transporter. As the mitochondria produce large quantities of ATP, then ATP needs to be shuffled out and transported into other parts of the cell. And so there, in this particular depiction, is showing the transporter on the inner mitochondrial membrane. Now I'm going to talk about one type of transporter, specifically that's a passive transporter. And as you can see, transporters move solutes along electrochemical gradients. And we're going to use the glucose transporter as our key example for this. So you want to know the details about how glucose transporter works. Just kind of FYI, the glucose transporter is embedded in a membrane. So you can see the cell membrane here. This is just a real kind of schematic look at the glucose transporter. It's actually a multi-pass transmembrane protein, and it has about 12 different passes across the membrane. So it's pretty impressive. But for now, what we're really interested in is how the transporter changes conformation as it is moving glucose across the membrane. So the big deal is that there are 3 conformations which the glucose transporter can use, and will go through as it moves glucose across the membrane. These are also called states rather than conformation in your textbook. And basically these states vary depending on the accessibility of the binding site. One of the keys about the glucose transporter is that it can switch randomly and reversibly between these conformations, and it does not depend on if it's bound or not. So here are the 3 different states. The first one is outward open. And you can see that our green blob, the glucose transporter, is open, facing the extracellular space. And in this case, you see the glucose is present outside of the cell. Again, this is going to open randomly. And so if glucose is present and happens to sneak in and bind at the binding site, then we'll go on to the next step. If there's no glucose that comes into the binding site, though, then it will still go on to the next state. So the second state is called the occluded state. Occluded is a word that means closed, and in other words, this particular state for the glucose transporter, it is closed or not open. And then again, whether or not the glucose is bound here, then we'll go into the next state for the glucose transporter. In this case, it is bound, and then it opens inwardly. We call this the inward open state. And if glucose is bound, that means then that as it opens inward, glucose can then dissociate and move into the cytoplasm. A really important point about how the glucose transporter works is that it moves glucose down its concentration gradient. And so the really critical part of understanding this transporter is, again, these rules about how it switches between the different conformational states. And so again, this is random and it's reversible, and it doesn't depend on the glucose binding or not. And so what that means is that if there's more glucose outside, then it's going to be more likely that the glucose will find the binding site when it's in the outward open state and then get moved across the membrane. And then you can also imagine this in the opposite fashion, where glucose, if it's at a low concentration, could still get pumped outside. However, this is just less likely to occur when there's a lower concentration of glucose inside the cell membrane. Something that's really important looking at these different transporters is we're really going to focus on how these events occur. So how this works: glucose will move with or down the concentration gradient through this glucose transporter. And basically wherever there's more glucose, it is more likely find the binding site and be transported across the membrane. So an example of this, I'm going to tell you about two different cell types and how in two different situations they're going to move glucose. So in this example, first we're going to talk about intestinal cells. And the situation in which someone has just eaten a meal, and so then there's very high concentration of glucose, which is outside the intestinal cell. The glucose transporter is going to function to move the glucose into the cell, down its concentration gradient, in this case. On the other hand, in liver cells, we're going to consider a different situation and concentration of glucose. And so for the liver cells, we're going to consider that someone's hungry and hasn't had any food for a while. This means there's going to be a low glucose concentration outside the cell. And then as a response, through some hormonal signaling, which we're not going to talk about, the liver cell will start to break down polysaccharides, such as glycogen, inside the cell, and then have a high concentration of glucose inside the cell. Glycogen, by the way, is a polysaccharide. And so what this means is that now there's a high concentration of glucose inside the cell. And basically, the purpose of this is to mobilize the glucose and get it out to other tissues. And so in this case, now the glucose transporter is going to work to move the glucose out of the cell. Again, this is with the concentration gradient.